1. Messenger RNA mRNA. It carries the genetic information copied from DNA in the form of a series of 3 base code words, each of which specifies a particular amino acid. RNA contains ribonucleotides of adenine, cytidine, guanine, and uracil. DNA contains deoxyribonucleotides of adenine, cytidine, guanine, and thymine. Because 4 nucleotides, taken individually, could represent only 4 of the 20 possible amino acids encoding the linear arrangement in proteins, a group of nucleotides is required to represent each amino acid. The code employed must be capable of specifying at least 20 words, i.e., amino acids. Synthesis of all protein chains in prokaryotic and eukaryotic cells begins with the amino acid methionine. In most mRNAs, the start initiator codon specifying this amino-terminal methionine is August. In a few bacterial mRNAs, GUG is used as the initiator codon, and CUG occasionally is used as an initiator codon for methionine in eukaryotes. The 3 codes AA, UGA, and UAG do not specify amino acids but constitute stop terminator signals that mark the carboxyl terminus of protein chains in almost all cells. The sequence of codons that runs from a specific start site to a terminating codon is called a reading frame. This precise array of row-nucleotides in groups of 3 in mRNA specifies the precise linear sequence of amino acids in a protein and also signals where synthesis of the protein chain starts and stops. 2. Transfer RNA tRNA. It is the key to deciphering the code words in mRNA. Each type of amino acid has its own type of tRNA, which binds it and carries it to the growing end of a polypeptide chain if the next code word on mRNA calls for it. The tRNA with its attached amino acid is selected at each step because each specific tRNA molecule contains a 3-base sequence that can base pair with its complementary code word in the mRNA. All tRNAs have 2 functions: to be chemically linked to a particular amino acid and to base pair with a codon in mRNA so that the amino acid can be added to a growing peptide chain. Each tRNA molecule is recognized by one and only one of the 20 amino acyl tRNA synthetases. Like each of these enzymes links one and only one of the 20 amino acids to a particular tRNA, forming an amino acyl tRNA. One correct acid is attached. A tRNA then recognizes a codon in mRNA, thereby delivering its amino acid to the growing polypeptide. As studies on tRNA proceeded, 30-40 different tRNAs were identified in bacterial cells and as many as 50,100 in animal and plant cells. The function of tRNA molecules, which are 70-80 nucleotides long, depends on their precise 3-dimensional structures. In solution, all tRNA molecules fold into a similar stem loop arrangement that resembles a cloverleaf when drawn in 2 dimensions. The ones are short double helices stabilized by Watson-Crick base pairing. 3 of the 4 stems have loops containing 7 or 8 bases at their ends, while the remaining, unlooped stem contains the free 3 and 5 ends of the chain. 3 nucleotides termed the anticodon, located at the center of one loop, can form base pairs with the 3 complementary nucleotides forming a codon in mRNA. As discussed later, specific amino acyl tRNA synthetases recognize the surface structure of each tRNA for a specific amino acid and covalently attach the proper amino acid to the unlooped amino acid acceptor stem. The 3 end of all tRNAs has the sequence CCA, which in most cases is added after synthesis and processing of the tRNA are complete. In 3 dimensions, the folded tRNA molecule has an L shape with the anticodon loop and acceptor stem forming the ends of the 2 arms. 3. Ribosomal RNA rRNA associates with a set of proteins to form ribosomes. These complex structures, which physically move along an mRNA molecule, catalyze the assembly of amino acids into protein chains. They also bind tRNAs and various accessory molecules necessary for protein synthesis. Ribosomes are composed of a large and small subunit, each of which contains its own rRNA molecule or molecules. H4. MicroRNA. In genetics, microRNAs mRNA, are single-stranded RNA molecules of about 21 to 23 nucleotides in length, which regulate gene expression. By genes that are transcribed from DNA but not translated into protein, non-coding RNA, instead they are processed from primary transcripts known as pre-mRNA to short stem loop structures called pre-mRNA and finally to functional mRNA. mRNA molecules are partially complementary to one or more messenger RNA mRNA molecules, and their main function is to down-regulate gene expression. They were first in 1993 by Lee and colleagues in the Victor Ambrose lab, yet the term microRNA was only introduced in 2001 in a set of 3 articles in Science, the 26th of October 2001. As of early 2008, computational analysis by IBM suggested the existence of as many as 50,000 different mRNAs in the typical mammalian cell, each with perhaps 1,000 or more potential target. Fig 1. The stem loop secondary structure of a pre-microRNA from Brassica oleracea. Formation and processing. The gene code is longer than the processed mature mRNA molecule. mRNAs are first transcribed as primary transcripts or pre-mRNA with a cap and poly a tail and processed to short, 70 nucleotide stem loop structures known as pre-mRNA in a cell nucleus. This processing is performed in animals by a protein complex known as the microprocessor complex, consisting of the nuclease DROCHA and the double-stranded RNA binding protein PASHA. These mRNAs are then processed to mature mRNAs in the cytoplasm by interaction with the endonuclease dicer, which also initiates the formation of the RNA-induced silencing complex RISC. This complex is responsible for the gene silencing observed due to mRNA expression and RNA interference. The pathway in plants varies slightly due to their lack of DROCHA homologs. Instead, dicer homologs alone affect several processing steps. The pathway is also different for mRNAs derived from intronic stem loops. These are processed by dicer but not by DROCHA. Either the sense strand or antisense strand of DNA can function as templates to give rise to mRNA. Efficient processing of pre-mRNA by DROCHA requires the presence of extended single-stranded RNA on both 3' and 5' ends of hairpin molecule. These ssRNA motifs could be of different composition while their length is of high importance if processing is to take place at all. A Informations of human and fly pre-mRNAs revealed very similar structural regions, called basal segments, lower stems, upper stems and terminal loops. Based on these conserved structures, thermodynamic profiles of pre-mRNA have been determined. The Drosha complex cleaves RNA molecule till the 2 helical turns away from the terminal loop and approximately 1 turn away from basal segments. In analyzed molecules, this region contains unpaired nucleotides and the free energy of the duplex is relatively high compared to lower and upper stem regions. Most pre-mRNAs don't have a perfect double-stranded RNA (dsRNA) structure topped by a terminal loop. There are few possible explanations for such selectivity. One could be that dsRNAs longer than 21 base pairs activate interferon response and antiviral machinery in the cell. Another plausible explanation could be that the thermodynamic profile of pre-mRNA determines which strand will be incorporated into dicer complex. Indeed, clear similarities between pre-mRNAs encoded in respective 5' or 3' strands have been demonstrated. Dicer cleaves the pre-mRNA stem loop. 2 complementary short RNA molecules are formed, but only one is integrated into the RISC complex. This strand is a guide strand and is selected by the Argonaute protein, the catalytically active RNAs in the RISC complex, on the basis of the stability of the 5' end. The remaining strand, known as the anti-guide or passenger strand, is degraded as a RISC complex substrate. After integration into the active RISC complex, mRNAs base pair with their complementary mRNA molecules and induce mRNA degradation by Argonaute proteins, the catalytically active members of the RISC complex. It is yet unclear how the activated RISC complex locates the mRNA targets in the cell, though it has been shown that the process is not coupled to ongoing protein translation from the mRNA. MicroRNA mRNA is produced from precursor microRNA, pre-mRNA, which in turn is formed from a microRNA primary transcript, pre-mRNA. Cellular functions. The function of mRNAs appears to be in gene regulation. For that purpose, a mRNA is complementary to a part of one or more messenger RNAs, mRNAs. mRNAs are usually complementary to a site in the 3' UTR, whereas plant mRNAs are usually complementary to coding regions of mRNAs. The annealing of the mRNA to the mRNA then inhibits protein translation, but sometimes facilitates cleavage of the mRNA. This is thought to be the primary mode of action of plant mRNAs. In such, the formation of the double-stranded RNA through the binding of the mRNA triggers the degradation of the mRNA transcript through a process similar to RNA interference, RNAi, though in other cases it is believed that the mRNA complex blocks the protein translation machinery or otherwise prevents protein translation without causing the mRNA to be degraded. mRNAs may also target methylation of genomic sites which correspond to targeted mRNAs. mRNAs function in association with a complement of proteins collectively termed the mRNP. This effect was first described for the worm C. elegans in 1993 by Victor Ambrose and coworkers. As of 2002, mRNAs have been confirmed in various plants and animals, including C. elegans, human and the plant Arabidopsis thaliana. University of Louisville has resulted in the production of microarrays containing all known, at the time of production, mRNAs for human, mouse, rat, dog, C. elegans and Drosophila species, tools referred to as chips. Agilent has subsequently commercialized a human mRNA microarray. Bacteria that are similar to eukaryotic mRNA genes in the sense that they control mRNA abundance or translation by binding an mRNA by base pairing, however they are not generally considered to be mRNAs because the dicer enzyme is not involved. Similar RNA species termed short interfering RNAs, siRNAs, are used to prevent the transcription of viral RNA. While this siRNA is double-stranded, the mechanism seems to be closely related to that of mRNA, especially taking the hairpin structures into account. siRNAs are also used to regulate cellular genes, as mRNAs do. Gene activation. dsRNA can also activate gene expression, a mechanism that has been termed small RNA-induced gene activation, or RNAA. dsRNAs targeting gene promoters can induce potent transcriptional activation of associated genes. This was demonstrated in human cells using synthetic dsRNAs termed small activating RNAs, SARNAs, but has also been demonstrated for endogenous microRNA. Detecting and manipulating mRNA signaling. The activity of an mRNA can be experimentally blocked using a locked nucleic acid oligo, a morpholino oligo, or a 2-pheto-methyl RNA oligo. Steps in the maturation of mRNAs can be blocked by steric blocking oligos. The target site of an mRNA on an mRNA can be blocked by a steric blocking oligo. mRNA and disease. Just as mRNA is involved in the normal functioning of eukaryotic cells, so has dysregulation of mRNA been associated with disease. This association in turn has led to increased funding opportunities for academic research and financial incentives for development and commercialization of mRNA-based diagnostics and therapeutics. Early commercialization aimed at academic research support was established. The initial research focus based on products and services requested was on cancer and neuroscience research. 2007, interests indicated by product and services requested broadened to include cardiac research, virology, cell biology in general, and plant biology. mRNA and cancer. Several mRNAs has been found to have links with some types of cancer. A study of mice altered to produce excess CMYC, a protein implicated in several cancers, shows that mRNA has an effect on the development of cancer. Mice that were engineered to produce a surplus of types of mRNA found in lymphoma cells developed the disease within 50 days and died 2 weeks later. In contrast, mice without the surplus mRNA lived over 100 days. Another study found that 2 types of mRNA inhibit the E2F1 protein, which regulates cell proliferation. mRNA appears to bind to messenger RNA before it can be translated to proteins that switch genes on and off. Measuring activity among 217 genes encoding mRNA, patterns of gene activity that can distinguish types of cancers can be discerned. mRNA signatures may enable classification of cancer. This will allow doctors to determine the original tissue type which spawned a cancer and to be able to target a treatment course based on the original tissue type. Profiling has already been able to determine whether patients with chronic lymphocytic leukemia had slow-growing or aggressive forms of the cancer. In 2008, the companies Asuragen and Exicon were working to commercialize this potential for mRNAs to act as cancer biomarkers. mRNA and heart disease. Of mRNA function in the heart has been addressed by conditionally inhibiting mRNA maturation in the murine heart, and has revealed that mRNAs play an essential role during its development. mRNA expression profiling studies demonstrate that expression levels of specific mRNAs change in diseased human hearts, pointing to their involvement in cardiomyopathies. Furthermore, studies on specific mRNAs in animal models have identified distinct roles for mRNAs both during heart development and under pathological conditions, including the regulation of key factors important for cardiogenesis, the hypertrophic growth response, and cardiac conductance. 2008, academic work on the relationship between mRNA and heart disease had advanced sufficiently to lead to the establishment of a company, Miragen, with a primary focus on cardiovascular health and disease. 1. Stability of proteins. The unique three-dimensional structure of each polypeptide is determined by its amino acid sequence. Interactions between the amino acid side chains guide the folding of the polypeptide to form a compact structure. 4 types of interactions cooperate in stabilizing the tertiary structures of globular proteins. 1. Disulfide bonds. A disulfide bond is a covalent linkage formed from a sulfhydryl group of each of 2 cysteine residues to produce a cysteine residue, figure 1. The 2 cysteines may be separated from each other by many amino acids in the primary sequence of a polypeptide, or may even be located on 2 different polypeptide chains. The folding of the polypeptide chains brings the cysteine residues into proximity, and permits covalent bonding of their side chains. A disulfide bond contributes to the stability of the three-dimensional shape of the protein molecule. For example, many disulfide bonds are found in proteins such as immunoglobulins that are secreted by cells. Note, these strong, covalent bonds help stabilize the structure of proteins, and prevent them from becoming denatured in the extracellular environment. 2. Hydrophobic interactions. Amino acids with nonpolar side chains tend to be located in the interior of the polypeptide molecule, where they associate with other hydrophobic amino acids, figure 2. In contrast, amino acids with polar or charged side chains tend to be located on the surface of the molecule in contact with the polar solvent. Note, these lipid environments, such as a membrane, exhibit the reverse arrangement. That is, hydrophilic amino acid side chains are located in the interior of the polypeptide, whereas hydrophobic amino acids are located on the surface of the molecule in contact with the nonpolar environment. In each case, the segregation of R groups occurs that is energetically most favorable. 3. Hydrogen bonds. Amino acid side chains containing oxygen or nitrogen-bound hydrogen, such as in the alcohol groups of serine and threonine, can form hydrogen bonds with electron-rich atoms, such as the oxygen of a carboxyl group or carbonyl group of a peptide bond. Formation of hydrogen bonds between polar groups on the surface of proteins in the aqueous solvent enhances the solubility of the protein, figure 3. 4. Alkanes. Negatively charged groups, such as the carboxyl group CO0 in the side chain of aspartate or glutamate, can interact with positively charged groups, such as the amino group NH3+ in the side chain of lysine. Folding. Interactions between the side chains of amino acids determine how long polypeptide chain folds into the intricate three-dimensional shape of the functional protein. Protein folding, which occurs within the cell in seconds to minutes, employs a shortcut through the maze of all folding possibilities. As a peptide folds, its amino acid side chains are attracted and repulsed according to their chemical properties. For example, positively and negatively charged side chains attract each other. Conversely, similarly charged side chains repel each other. In addition, interactions involving hydrogen bonds, hydrophobic interactions, and disulfide bonds all seek to exert an influence on the folding process. This process of trial and error tests many, but not all, possible configurations, seeking a compromise in which attractions outweigh repulsions. This results in a correctly folded protein with a low energy state. Role of chaperones in protein folding. It is generally accepted that the information needed for correct protein folding is contained in the primary structure of the polypeptide. Given this, it is difficult to explain why most proteins when denatured do not resume their native conformations under favorable environmental conditions. One answer to this problem is that a protein begins to fold in stages during its synthesis, rather than waiting for synthesis of the entire chain to be totally completed. This limits competing folding configurations made available by longer stretches of nascent peptide. In addition, a specialized group of proteins, named chaperones, are required for the proper folding of many species of proteins. The chaperones, also known as heat shock proteins, interact with the polypeptide at various stages during the folding process. Some chaperones are important in keeping the protein unfolded until its synthesis is finished, or act as catalysts by increasing the rates of the final stages in the folding process. Others protect proteins as they fold so that their vulnerable, exposed regions do not become tangled in unproductive encounters. Quaternary structure of protein. Many proteins consist of a single polypeptide chain, and are defined as monomeric U proteins. However, others may consist of 2 or more polypeptide chains that may be structurally identical or totally unrelated. The arrangement of these polypeptide subunits is called the quaternary structure of the protein. Note, if there are 2 subunits, the protein is called dimeric, if 3 subunits, trimeric, and, if several subunits, multimeric. Subunits are held together by noncovalent interactions, for example, hydrogen bonds, ionic bonds, and hydrophobic interactions. Subunits either function independently of each other, or may work cooperatively, as in hemoglobin, in which the binding of oxygen to one subunit of the tetramer increases the affinity of the other subunits for oxygen. Denaturation of protein. Protein denaturation results in the unfolding and disorganization of the protein's secondary and tertiary structures, which are not accompanied by hydrolysis of peptide bonds. Denaturing agents include heat, organic solvents, mechanical mixing, strong acids or bases, detergents, and ions of heavy metals such as lead and mercury. Denaturation may, under ideal conditions, be reversible, in which case the protein refolds into its original native structure when the denaturing agent is removed. However, most proteins, once denatured, remain permanently disordered. Denatured proteins are often insoluble and, therefore, precipitate from solution. 2. Stability of NuClIEC acids. In the Watson-Crick structure, the 2 chains or strands of the helix are antiparallel. Their 5' 3' phosphodiester bonds run in opposite directions. Work with DNA polymerases provided experimental evidence, confirmed by X-ray crystallography, that the strands are indeed antiparallel. To account for periods observed in the X-ray diffraction pattern, Watson and Crick used molecular models to show that the vertically stacked bases inside the double helix would be 0.34 nanometers apart and that the secondary repeat distance of about 3.4 nanometers could be accounted for by the presence of 10, now 10.5 nucleotide residues in each complete turn of the double helix. The antiparallel polynucleotide chains of double helical DNA are not identical in either base sequence or composition. Instead, they are complementary to each other. Wherever adenine appears in one chain, thymine is found in the other. Similarly, wherever guanine is found in one chain, cytosine is found in the other. DNA double helix or duplex is held together by two sets of forces, as described earlier, hydrogen bonding between complementary base pairs and base stacking interactions. The specificity that maintains a given base sequence in each DNA strand is contributed entirely by the hydrogen bonding between base pairs. Base stacking interactions, which are largely nonspecific with respect to the identity of the stacked bases, make the major contribution to the stability of the double helix. J1. Metabolism of CARBOHYDRARES, citric acid cycle. The citric acid cycle, Krebs cycle or tricarboxylic acid TCA cycle, is the most important metabolic pathway for the energy supply to the body. About 65–70% of the ATP is synthesized in Krebs cycle. Citric acid cycle essentially involves the oxidation of acetyl-CoA to CO2 and H2O. The cycle utilizes about 2/3 of total oxygen consumed by the body. The TCA cycle is used, since, at the outset of the cycle, tricarboxylic acids, citrate, cis-aconitate and isocitrate participate. TCA cycle, the central metabolic pathway. The citric acid cycle is the final common oxidative pathway for carbohydrates, fats and amino acids. This cycle not only supplies energy but also provides many intermediates required for the synthesis of amino acids, glucose, heme, etc. Krebs cycle is the most important central pathway connecting almost all the individual metabolic pathways either directly or indirectly. Briester, the citric acid cycle was proposed by Hans Adolf Krebs in 1937, based on the studies of oxygen consumption in pigeon breast muscle. The cycle is named in his honor and he was awarded the Nobel Prize for Physiology and Medicine in 1953. Location of TCA cycle. The enzymes of TCA cycle are located in mitochondrial matrix, in close proximity to the electron transport chain. This enables the synthesis of ATP by oxidative phosphorylation without any hindrance. TCA cycle in overview. Cycle basically involves the combination of a 2-carbon acetyl-CoA with a 4-carbon oxaloacetate to produce a 6-carbon tricarboxylic acid, citrate. In the reactions that follow, the 2 carbons are oxidized to CO2 and oxaloacetate is regenerated and recycled. Oxaloacetate is considered to play a catalytic role in citric acid cycle. TCA cycle in open cycle. Krebs cycle is a cyclic process. However, it should not be viewed as a closed circle, since many compounds enter the cycle and leave. TCA cycle is comparable to a heavy traffic circle in a national highway with many connecting roads. Each intermediate of the cycle connecting another pathway is a road. Reactions of citric acid cycle. Oxidative decarboxylation of pyruvate to acetyl-CoA by pyruvate dehydrogenase complex is discussed above. This step is a connecting link between glycolysis and TCA cycle. A few authors, however, describe the conversion of pyruvate to acetyl-CoA along with citric acid cycle. The events of TCA cycle are described hereunder. Step 1. Formation of citrate. Krebs cycle proper starts with the condensation of acetyl-CoA and oxaloacetate, catalyzed by the enzyme citrate synthase. This aldol condensation reaction leading to the formation of an intermediate citral-CoA, which, on hydrolysis, yields citrate. Citrate is freely permeable across the mitochondrial membrane. It serves as a good source of cytosolic acetyl-CoA which is used for the synthesis of fatty acids. Citrate inhibits phosphofructokinase and activates acetyl-CoA carboxylase. The latter is the key enzyme in fatty acid synthesis. Step 2 and 3. Citrate is isomerized to isocitrate by the enzyme aconitase. This is achieved in a two-stage reaction of dehydration followed by hydration through the formation of an intermediate cis-aconitate. Step 4 and 5. Formation of alpha-ketoglutarate. The enzyme isocitrate dehydrogenase LCD catalyzes the conversion oxidative decarboxylation of isocitrate to oxalosuccinate and then to acetoglutarate. The formation of NADH and the liberation of C02 occurs at this stage. 3 types of ICD are known. NAD plus specific, located only in mitochondria. NADP plus specific present in mitochondria, and cytosol 3. The mitochondrial NAD plus dependent ICD is responsible for the conversion of isocitrate to alpha-ketoglutarate. Step 6. Conversion of alpha-ketoglutarate to succinyl-CoA. Occurs through oxidative decarboxylation, catalyzed by acetoglutarate dehydrogenase complex. This enzyme is dependent on 5 cofactors TPP, lipoamide, NAD+, fat and CoA. The mechanism of the reaction is analogous to the conversion of pyruvate to acetyl-CoA. At this stage of the TCA cycle, second NADH is produced and the second CO2 is liberated. Step 7. Formation of succinate. Succinyl-CoA is converted to succinate by succinate thiokinase. This reaction is coupled with the phosphorylation of GDP to GTP. This is a substrate-level phosphorylation. GTP is converted to ATP by the enzyme nucleoside diphosphate kinase. GTP plus ADP left pointing arrow right pointing arrow ATP plus GDP. Step 8. Conversion of succinate to fumarate. Succinate is oxidized by succinate dehydrogenase to fumarate. This reaction results in the production of FADH2 and not NADH. This is due to the fact that the reducing power of succinate is not adequate to reduce NAD+, hence fat is utilized. Step 9. Formation of malate. The enzyme fumarase catalyzes the conversion of fumarate to malate with the addition of H2O. Step N. Conversion of malate to oxaloacetate. Malate is then oxidized to oxaloacetate by malate dehydrogenase. The third and final synthesis of NADH occurs at this stage. Oxaloacetate is regenerated, which can combine with another molecule of acetyl-CoA and continue the cycle. Summary of TCA cycle. The events of Krebs cycle may be summarized as acetyl plus 3 Namibian dollars plus FAD plus GDP plus pi plus 2H2O2 CO2 plus 3 NADH plus 3H plus FADH2 plus GTP plus CoA. Requirement of O2 by TCA cycle. There is no direct participation of oxygen in Krebs cycle. However, the cycle operates only under aerobic conditions. This is due to that NAD plus and FAD from NADH and FADH2 respectively required for the operation of the cycle can be regenerated in the respiratory chain only in the presence of O2. Citric acid cycle is strictly aerobic in contrast to glycolysis, which operates in both aerobic and anaerobic conditions. Citric acid cycle. During the process of oxidation of acetyl-CoA via citric acid cycle, 4 reducing equivalents 3 as NADH and 1 as FADH2 are produced. Oxidation of 1 NADH by electron transport chain coupled with oxidative phosphorylation results in the synthesis of 3 ATP, whereas FADH2 leads to the formation of 2 ATP. Besides, there is 1 substrate level phosphorylation. A total of 12 ATP are produced from 1 acetyl-CoA. Inhibitors of Krebs cycle. The important enzymes of TCA cycle inhibited by the respective inhibitors are listed. Enzyme inhibitor. Aconitase fluoroacetate, non-competitive alpha. Ketoglutarate arsenite, non-competitive dehydrogenase. Succinate dehydrogenase malonate, competitive regulation of citric acid cycle. The cellular demands of ATP are crucial in controlling the rate of citric acid cycle. The regulation is brought about either by enzymes or the levels of ADP. 3 enzymes, namely citrate synthase, isocitrate dehydrogenase and alpha-ketoglutarate dehydrogenase regulate citric acid cycle. 1. Citrate synthase is inhibited by ATP, NADH, acyl-CoA and succinyl-CoA. 2. Isocitrate dehydrogenase is activated by ADP and inhibited by ATP and NADH. 3. Alpha-ketoglutarate dehydrogenase is inhibited by succinyl-CoA and NADH. 4. Availability of ADP is very important for the citric acid cycle to proceed. This is due to the fact that unless sufficient levels of ADP are available, oxidation coupled with phosphorylation of ADP to ATP, of NADH and FADH2 through electron transport chain stops. The inhalation of NADH and FADH2 will lead to inhibition of the enzymes, as stated above, and also limits the supply of NAD plus and FAD, which are essential for TCA cycle to proceed. Amphibilic nature of the citric acid cycle. The citric acid cycle is the final common oxidative pathway in the living cells. The cycle also provides various intermediates for the synthesis of many compounds needed by the body. Krebs cycle is both catabolic and anabolic in nature, hence regarded as amphibilic. The most important synthetic anabolic reactions connected with TCA cycle are given. 1. Oxaloacetate and alpha-ketoglutarate, respectively, serve as precursors for the synthesis of aspartate and glutamate, which, in turn, were required for the synthesis of other non-essential amino acids, purines and pyrimidines. 2. Succinyl-CoA is used for the synthesis of porphyrins and heme. 3. Mitochondrial citrate is transported to the cytosol, where it is cleaved to provide acetyl-CoA for the biosynthesis of fatty acids, sterols, etc. Energetics of glucose oxidation. When a molecule of glucose, 6-carbon, undergoes glycolysis, 2 molecules of pyruvate or lactate, 3-carbon, are produced. Pyruvate oxidatively decarboxylated to acetyl-CoA, 2-carbon, which enters the citric acid cycle and gets completely oxidized to CO2 and H2O. The overall process of glucose being completely oxidized to CO2 and H2O via glycolysis and citric acid cycle is C6H12O6 plus 6O2 plus 38 ADP plus 38 pi 6 CO2 plus 6 H2O plus 38 ATP. When a molecule of glucose is burned in a calorimeter, 2,780 kilojoules of heat is liberated. In the living this energy is trapped, leading to the synthesis of 38 ATP, which is equivalent to 1,159 kilojoules. 1 ATP has high energy bond equivalent to 30.5 kilojoules. That is, about 48% of the energy in glucose combustion is actually captured for ATP generation. J2. Gluconeogenesis. The synthesis of glucose or glycogen from non-carbohydrate compounds is known as gluconeogenesis. The major substrates precursors for gluconeogenesis are lactate, pyruvate, glucogenic amino acids, propionate and glycerol. Location of gluconeogenesis. Gluconeogenesis occurs mainly in the cytosol, although some precursors are produced in the mitochondria. Gluconeogenesis mostly takes place in liver and, to some extent, in kidney matrix, about 1/10 of liver capacity. Import of gluconeogenesis. Glucose occupies a key position in the metabolism and its continuous supply is absolutely essential to the body for a variety of functions. 1. Central nervous system, or erythrocytes, testes and kidney medulla are dependent on glucose for continuous supply of energy. Human brain alone requires about 120 grams of glucose per day, out of about 160 grams needed by the entire body. 2. Glucose is the only source that supplies energy to the skeletal muscle, under anaerobic conditions. 3. All the carbohydrates are synthesized from glucose, e.g. lactose, amino sugars, etc. 4. Glucose is converted to glycerol and utilized for the synthesis of fat. 5. In fasting even more than a day, gluconeogenesis must occur to meet the basal requirements of the body for glucose and to maintain the intermediates of citric acid cycle. This is essential for the survival of humans and other animals. 6. Certain metabolites produced in the tissues accumulate in the blood, e.g. lactate, glycerol, propionate, etc. Gluconeogenesis effectively clears them from the blood. Reactions of gluconeogenesis. Gluconeogenesis closely resembles the reverse pathway of glycolysis, although it is not the complete reversal of glycolysis. Essentially, 3 out of 10 reactions of glycolysis are irreversible. The 7 reactions are common for both glycolysis and gluconeogenesis Fig. The irreversible steps of glycolysis are catalyzed by the enzymes, namely hexokinase, phosphofructokinase and pyruvate kinase. These 3 stages bypassed by alternate enzymes specific to gluconeogenesis are discussed. 1. Conversion of pyruvate to phosphoenol pyruvate. This takes place in 2 steps. Pyrocarboxylase is a biotin-dependent mitochondrial enzyme that converts pyruvate to oxaloacetate in presence of ATP and CO2. This enzyme regulates gluconeogenesis and requires acetyl-CoA for its activity. Oxaloacetate is synthesized in the mitochondrial matrix. It has to be transported to the cytosol to be used in gluconeogenesis, where the rest of the pathway occurs. Due to membrane impermeability, oxaloacetate cannot diffuse out of the mitochondria. It is converted to malate and then transported to the cytosol. Within the cytosol, oxaloacetate is regenerated. Reversible conversion of oxaloacetate and malate is catalyzed by malate dehydrogenase, an enzyme present in both mitochondria and cytosol. In the cytosol, phosphoenol pyruvate carboxykinase converts oxaloacetate to phosphoenol pyruvate. ATP or ITP, not ATP, is used in this reaction and the CO2, fixed by carboxylase, is liberated. For the conversion of pyruvate to phosphoenol pyruvate, 2 ATP equivalents are utilized. This is in contrast to only 1 ATP that is liberated in glycolysis for this reaction. 2. Conversion of fructose 1,6-bisphosphate to fructose. 6-phosphate, phosphoenol pyruvate undergoes the reversal of glycolysis until fructose 1,6-bisphosphate is produced. The enzyme fructose 1,6-bisphosphatase converts fructose 1,6-bisphosphate to fructose 6-phosphate. This enzyme requires Mg2+ ions. Fructose 1,6-bisphosphatase is absent in smooth muscle and hard muscle. This enzyme is also regulatory in gluconeogenesis. 3. Conversion of glucose 6-phosphate to glucose. Glucose 6-phosphatase catalyzes the conversion of glucose 6-phosphate to glucose. The presence or absence of this enzyme in a tissue determines whether the tissue is capable of contributing glucose to the blood or not. It is mostly present in liver and kidney but absent in muscle, brain and adipose tissue. The overall summary of gluconeogenesis for the conversion of pyruvate to glucose is shown. Pyruvate plus 4 ATP plus 2 GTP plus 2 NADH plus 2 H plus 6 H2O. Glucose plus 2 Namibian dollars plus 4 ADP plus 2 GDP plus 6 Pi plus 6 H plus. After the formation of glucose, its conversion to glycogen occurs through the formation of uridine diphosphate glucose, UDP glucose, by the enzyme glycogen synthetase. Gluconeogenesis from amino acids. The carbon skeleton of glucogenic amino acids, all except leucine and lysine, results in the formation of pyruvate or the intermediates of citric acid cycle which, ultimately, result in the synthesis of glucose. Gluconeogenesis from glycerol. Glycerol is liberated mostly in the adipose tissue by the hydrolysis of fats triacylglycerols. The enzyme glycerokinase, found in liver and kidney, absent in adipose tissue, activates glycerol to glycerol 3-phosphate. The latter is converted to dihydroxyacetone phosphate by glycerol 3-phosphate dehydrogenase. Dihydroxyacetone phosphate is an intermediate in glycolysis which can be conveniently used for glucose production. Gluconeogenesis from propionate. Oxidation of odd-chain fatty acids and the breakdown of some amino acids methionine, isoleucine, yields a 3-carbon propionyl-CoA. Propionyl-CoA carboxylase acts on this in presence of ATP and biotin and converts to methylmalonyl-CoA which is then converted to succinyl-CoA in presence of B12 coenzyme. Succinyl-CoA formed from propionyl-CoA enters gluconeogenesis via citric acid cycle. Propionate is an important precursor for gluconeogenesis in ruminant animals such as cattle. Propionate is activated to propionyl-CoA which is then converted to glucose as described above. Gluconeogenesis from lactate Cori cycle. Lactate produced by active skeletal muscle is a major precursor for gluconeogenesis. Under anaerobic conditions, pyruvate is reduced to lactate by lactate dehydrogenase LDH. LDH. Pyruvate plus NADH plus H plus lactate plus NAD plus. Lactate is a dead end in glycolysis, since it must be reconverted to pyruvate for its further metabolism. The very purpose of lactate production is to regenerate NADH so that glycolysis proceeds uninterrupted in skeletal muscle. Lactate or pyruvate produced in the muscle cannot be utilized for the synthesis of glucose due to the absence of the key enzymes of gluconeogenesis, glucose 6-phosphatase and fructose 1,6-bisphosphatase. The plasma membrane is freely permeable to lactate. Lactate is carried from the skeletal muscle through blood and handed over to liver, where it is oxidized to pyruvate. Pyruvate, so produced, is converted to glucose by gluconeogenesis, which is then transported to the skeletal muscle. Thus involving the synthesis of glucose in liver from a skeletal muscle lactate and the reuse of glucose thus synthesized by the muscle for energy purposes known as Cori cycle Fig. Glucose-alanine cycle. There is a continuous transport of amino acids from muscle to liver, which predominantly occurs during starvation. Alanine dominates among the amino acids. Alanine dominates among the amino acids. It is postulated that pyruvate in skeletal muscle undergoes transamination to produce alanine. Alanine is transported to liver and used for gluconeogenesis. This cycle is referred to as glucosylanine cycle. Regulation of gluconeogenesis. The hormone glucagon and the availability of substrates mainly regulate gluconeogenesis, as discussed here under. 1. Influence of glucagon. This is a hormone, secreted by A cells of the pancreatic islets. Glucagon stimulates gluconeogenesis by 2 mechanisms. A. Active form of pyruvate kinase is converted to inactive form through the mediation of cyclic AMP, brought about by glucagon. Decreased pyruvate kinase results in the reduced conversion of phosphoenol pyruvate to pyruvate and the former is diverted for the synthesis of glucose. B. Glucagon reduces the concentration of fructose 2,6-bisphosphate. This compound allosterically inhibits phosphofructokinase and activates fructose 1,6-bisphosphatase, both favor increased gluconeogenesis. Generally, when gluconeogenesis is more active, glycolysis is very low. This is a good example of cellular economy. 2. Availability of substrates. Among the various substrates, glucogenic amino acids have stimulating influence on gluconeogenesis. This is particularly important in a condition like diabetes mellitus, decreased insulin level, where amino acids are mobilized from muscle protein for the purpose of gluconeogenesis. 3. Acetyl-CoA promotes gluconeogenesis. During starvation due to excessive lipolysis in adipose tissue, acetyl-CoA accumulates in the liver. Acetyl-CoA allosterically activates pyruvate carboxylase, resulting in enhanced glucose production. Gluconeogenesis from fat. It is often stated that glucose cannot be synthesized from fat. It is certainly true, since the fatty acids, most of them being even chain, on oxidation, produce acetyl-CoA which cannot be converted to pyruvate. Further, the 2 carbons of acetyl-CoA disappear as 2 moles of CO2 in TCA cycle. Therefore, even chain fatty acids cannot serve as precursors for glucose formation. However, the glycerol released from lipolysis and the propionate obtained from the oxidation of odd-chain fatty acids are good substrates for gluconeogenesis, as discussed above. J3. Glycogen metabolism. Glycogen is the storage form of glucose in animals, as is starch in plants. It is stored mostly in liver, 6-8%, and muscle, 1-2%. Due to more muscle mass, the quantity of glycogen in muscle, 250 grams, is about 3 times higher than that in the liver, 75 grams. Functions of glycogen. The prime function of liver glycogen is to maintain the blood glucose levels, particularly between meals. Liver glycogen stores increase in a well-fed state which are depleted during fasting. Muscle glycogen serves as a fuel reserve for the supply of ATP during muscle contraction. Structure of glycogen in brief. Glycogen is a homopolysaccharide composed of alpha-D glucose, up to 100,000 residues may be present. The glucose units are held together by alpha-1, 4 linkages. After 8 to 10 residues of glucose, a branch is formed with alpha-1, 6 glycosidic linkage. Glycogen is stored as granules in the cytosol, where most of the enzymes of glycogen synthesis and breakdown are present. Glycogenesis. The synthesis of glycogen from glucose is glycogenesis, Fig. Glycogenesis takes place in the cytosol and requires ATP and UTP, besides glucose. 1. Synthesis UDP glucose. The enzymes hexokinase in muscle and glucokinase in liver convert glucose to glucose 6-phosphate. Phosphoglucomatase catalysis the conversion of glucose 6-phosphate to glucose 1-phosphate. Pyridine diphosphate glucose UDPG is synthesized from glucose 1-phosphate and UTP by UDP glucose pyrophosphorylase. Pyrophosphate PPI produced in this reaction is hydrolyzed to inorganic phosphate pi by pyrophosphatase. This will ensure the optimal synthesis of UDPG. 2. Required primer to initiate glycogenesis. A small fragment of pre-existing glycogen must act as a primer to initiate glycogen synthesis. It is recently found that in the absence of glycogen primer, a specific protein namely glycogenin can accept glucose from UDPG. The hydroxyl group of the amino acid tyrosine of glycogenin is the site at which the initial glucose unit is attached. The enzyme glycogen initiator synthase transfers the first molecule of glucose to glycogenin. Then glycogenin itself takes up a few glucose residues to form a fragment of primer which serves as an acceptor for the rest of the glucose molecules. 3. Glycosynthesis by glycogen synthase. Glycogen